Just going to give the link to Twitter and Telegram again. Sorry, guys. I'm going to be right back at this. The juice reduces transmission. The juice reduces transmission the way that delayed onset gasoline puts out a fire. So, say, for example, that a new company says to the fire department, we have this new product that puts out gasoline better than water, and we want you to try it out. It's called delayed onset gasoline. It smothers the heck out of fire, and it doesn't ignite into flames until 3 hours later. So they do a study. They put the cutoff for the data collection at 2 hours, and they show that it's exceedingly good at putting out the house fire. It puts it out faster than the water does. And because they didn't go to the 3rd hour, they didn't see that later the house exploded. And so the analogy here is that based on the 5 studies that I'm going to show you, and if you're on Instagram, I gave you, well, if you're anywhere, the shortcuts are chrismasterjohnphd.com/juice, and then add the number 1, 2, 3, 4, or 5. Each one of those 5 different links will bring up the 5 studies that we're going to talk about today. On YouTube and Facebook, those links are in the description. You can just click on them. And these are 5 studies that overall what they show is that the juice had a transient several month transmission reducing effect during the dominance of the pre-delta phases. For example, the juice reduced transmission during the alpha variant. And by doing that, it allowed to take over the delta variant, whose transmission it cannot offer any protection against transmission of at all. And so because it reduced the transmission of the alpha variant in favor of the delta, that is why delta spread so fast during that time period. And now we are left with delta, whose transmission is not protected against by the juice, except possibly for a very, very, very short time frame after being juiced that is way shorter than any of the additional juices that people will get in those intervals that follow. So it's not 6 months. It's not 12 months. It's, probably a maximum 2-month position, such that if you were to try to filter out the population between who's juiced and who's unjuiced, it's not going to tell you a darn thing about whether they have any protection of transmission against delta, because they probably have none or close to none. And what we're going to see is if they have no symptoms, it's possible that they are more likely to transmit delta. And so does the juice reduce transmission? Well, that's contextual. It seems to have reduced transmission in the past of the things that whose transmission it can protect against, and by doing so cause to take over the things whose transmission it cannot protect against. And now we're left with delta, and we're left with the juice doing nothing. And only now that we are in that position do we now have mandates saying you must be juiced or you get fired. You must be juiced or you can't come in here. Now that we are in the situation that only took a few months to occur, in which whether someone's juiced or unjuiced tells us nothing about whether they are transmissive or not. And so let's now take a look at those studies. So I'm going to screen share on YouTube, but if you're not on YouTube, you can go to chrismasterjohnphd.com/juice1 to look at the first study. So I'm going to share my screen, and just to make sure that my link is working for anyone who says that, we go to chrismasterjohnphd.com/juice1, and it brings up the study that we're talking about. Now, this study is very small. It was done at the University of Illinois, and it was pre-Delta. So keep in mind that the claims that they're making in this study only apply pre-Delta. And if we click on info in history, we can see that most of the authors are from University of Illinois at Urbana-Champaign, or from Northwestern University, which is nearby, or from the University of Chicago School of Medicine. One author is from the Theoretical Biology and Biophysics Los Alamos National Laboratory Lab in New Mexico. And let's see quickly. I forget where they put funding in here. So let's see who funded it. Probably in the full text. If we keyword search for funding, this work was supported by the National Heart, Lung, and Blood Institute at the NIH. And it was also supported by a Northwestern University grant. All right, so it was supported by NIH and a Northwestern University grant, and this isn't industry funded or funded by any kind of think tank or anything like that. Okay, so if we go to the abstract, just to get a broad overview of what they found. Now, this is a preprint. By the way, there exists no studies. First of all, I should clarify. The 5 studies that I've selected are the 5 that I can find so far, and this might be all of them. But they're the 5 that I found so far that attempted to culture virus from a swab, and essentially people's snot, and or in this case, I think they also did it from saliva, and so some people would question whether they're culturing the virus. They're not really isolating the virus, but what they're doing is they're showing that if you drop the sample on some cells, you will observe a cytopathic effect, meaning you cause toxicity to the cells. And then they basically run a PCR test on the cells to show that there is RNA from the virus being replicated. And so it replicates and it's causing toxicity, and its RNA is the same as what you're testing for. And so it sort of looks and smells like a virus, but they're not like isolating the virus, but it still looks and smells and otherwise behaves like a virus because it replicates, because it's based on RNA, because it matches the positive test, and because it causes toxicity to the cells. So when they see those things align, that's what they're calling culturing the virus or isolating the virus. And this is widely used as a key metric to indicate that someone would be infectious or transmissive, right? Because remember, we're not talking about does someone get infected here. What we want to know is, we're not talking about whether someone gets a positive test. We're asking the question, if you take their snot and you look for the virus, do you find replicating virus that would appear to be infective towards other people? Because that's what matters when you're talking about transmission. Now, keep in mind, I'm not looking at secondary attack rates and stuff. So there's more evidence on the topic than what I'm covering here. But what I'm covering here is limited to studies showing infectious virus isolation, so-called, from people's snot. Okay, so what they say in the abstract is the durations of both infectious virus shedding and symptoms were significantly reduced in vaccinated individuals compared with unvaccinated. We also observed that breakthrough infections are associated with strong tissue compartmentalization and are only detectable in saliva in some cases. These data indicate that this data indicates that the juice shortens the duration of time of high transmission potential, minimizes symptom duration, and may restrict tissue dissemination. So one of the things that's notable is that there's not that many people in this study. And so this was at 2 universities, University of Illinois, Urbana-Champaign, where I did my postdoc, where I used to teach in the veterinary vitamins and minerals in the veterinary school, and Northwestern University. And this is during a time where I am not sure, but I think there was a juice mandate at the schools, because when they're comparing to unjuiced people, they are not using people that were sampled during this cohort. They're using a previously published sample. So this study is really terrible at comparing juiced and unjuiced people, because the unjuiced people aren't even from the same time period. I don't know why they would do that, except that I think they didn't have any unjuiced people during this period because they probably mandated the juice. And so what they did was they depending on the there were two universities. So in one case they were doing saliva testing with PCR. Another case they were doing lamp testing, which is a little bit different. And so the sampling isn't exactly the same, but it's preemptive sampling to try to screen, where every 2 to 4 days they have to get tested. And if they test positive, they have 24 hours to be part of this study. And in some cases, their contacts had 24 hours or a few days to decide to be part of this study as well. And so those people are separated into whether they've been fully juiced or whether they've been newly juiced, which means that they got their second juice or their final juice if it was, or their only juice if it was J&J. But it hasn't been 2 weeks since. So fully juiced is after 2 weeks after the final juice dose, and newly juiced is you've gotten the final juice dose, but it hasn't been 2 weeks yet. And then partially juiced is you haven't gotten the final juice dose yet. So you've gotten one dose of juice, but you haven't gotten the second one, if it applies. And so one of the things that we can see here is in figure 1, they're plotting out the individual numbers of people. And so the fully juiced people have, there's 1, 2, 3, 4, 5, 6 fully juiced people. The partially juiced is 1, 2, 3, 4, 5, 6 people. And the newly juiced are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 people. So there's almost no one in this study. And then they compare to unjuiced people from a different time period, because they didn't have any unjuiced people from that time period at the school. And it's all pre-Delta. So let me see if let me see where they describe the variance. So one second, I'm just looking for the right. I think they actually. Okay, here's what it is. So they say these individuals were primarily infected with alpha and gamma variants, as enrollment in the study concluded before the widespread circulation of Delta at the study sites. So they're telling you directly here, this is pre-Delta. And so like I was saying, pre-Delta, there was a transmission reducing effect. But so what this I don't want to go into detail on what this study is showing, because I'm making the point that it's not relevant anymore. And so let's just rely on the abstract so that the video doesn't wind up getting so long. But what they're saying is that in a small sample of people who were not properly controlled to unjuiced, where the sample size was 6 to 12 people in each category, the durations of the infectious shedding and symptoms were significantly reduced in the juiced people compared to the unjuiced people from a different time period, a different cohort, not properly controlled. And there was strong tissue compartmentalization, which means that, and what they mean by that is that if they tested positive in saliva, it seemed to kind of go up and down in the saliva and never make it to the nose, except in one person where it passed up to the nose. And what that indicates to me is that the saliva testing is overly sensitive and it's catching people who are infected and are irrelevant. That's what that means to me. And so the number of people who are actually positive in any real way from the nose is even smaller. So the saliva tested people in here are not even relevant. So this is pre-Delta, not properly controlled, very poor sample size, but it's worth noting because it's one of the 5 studies that did culture for the virus. Okay, so now we're going to go to chrismasterjohnphd.com/juice2, and I'm just going to make sure I get the same study if I type that in for the people who aren't seeing screen share. And it indeed pulls it up. Okay, so this is a preprint, and that means it's not peer-reviewed yet. That's true of every single one of the studies that we're looking at so far. But what does that tell you? That there aren't any peer-reviewed studies looking at whether the juice reduces the amount of infectious virus in anyone's nose. Right? What does that mean? Okay, so so just briefly, the authors are from Johns Hopkins School of Medicine, National, so NIAID of NIH, like the, where Fauci's from. Fauci's not on this, but it's, it's from the NIAID. And just those two. So Johns Hopkins and NIAID. Let's look at the funding. Funding came from NIH, grant to the Johns Hopkins Center of Excellence in Influenza Research and Surveillance, Johns Hopkins University, Maryland Department of Health, and CDC. All right. So NIH, Johns Hopkins, Maryland Department of Health, CDC. So no think tanks, no industry, etc. All right. So this paper, now, this paper, along with the next paper, which came from the same group of authors at two different time periods, are the key to this concept of the delayed onset gasoline. Remember, the analogy I'm making is that the juice reduces transmission, like delayed onset gasoline puts out a fire. And you can sell that if you look at the delayed onset gasoline smothering the fire, if you don't wait for the house to explode after the gasoline ignites. All right. And so we're going to look at two studies from the same group of authors with the same funding and everything in the same place, doing the same thing, who showed one thing during alpha and then a different thing during delta. And actually in the second paper compared alpha and delta. And that's the critical comparison that we're going to make. Okay. So, so the title of this paper is Infections in Juiced Individuals Are Biased Towards Viruses Encoding for Spike E484K mutation and associated with reduced infectious viral loads that correlate with respiratory antiviral IgG. Now, this Spike E484K, as far as I can tell, and I didn't deep dive into this, but it looked to me like this is a mutation that is generally associated with Delta, but can also be found in Iota or Iota variants. But what's notable is that they're finding a bias towards this mutation in the juiced people compared to the unjuiced people, which shows you selection for that mutation, right? Because if that mutation helps get around the immunity, and remember, juiced immunity is narrow compared to natural immunity because it's focused on spike. And so you would completely expect it, and it would defy logic if it didn't select for mutations in spike that avoid the juice immunity. And so they're actually providing a hint here before they do their next study, actually showing the fact that the juice is currently selecting, all right? It's selecting for mutations that get around the juice. All right, so in the abstract, they say, in the study, we had 133 positive patients who had two doses of either of the two main juices, the second of which was received between January and April of 2021. Positive samples were collected between January and May of 2021. In that time, extending from 2 to 100 days after the second dose of juice, samples were sequenced to characterize the whole genome and spike protein changes and cycle thresholds that reflect viral loads with a single molecular assay that they used. It's not totally relevant to go into the details. Of 133 positive tests, 24 couldn't be sequenced. Of 109, 2/3 of them were from symptomatics. 10% of them were admitted to the hospital, and 2% of them required ICU, but none of them were associated with mortality. The predominant strain was alpha. However, there was a significant association between one of the Iota lineages and the spike E484K mutation, with positives after being juiced when they were compared to a large control cohort that was matched for the time frame. A significant reduction of the recovery of infectious virus on cell culture, as well as delayed time to first appearance of cytopathic effect. Because remember, when they culture the virus, what they're doing is they're dumping the sample on the cells, waiting for them to get hurt, cytopathic effect, that's called, and then testing them for the RNA and showing that RNA of the virus is being replicated, right? So this is the people who say the virus hasn't been replicated. They're pointing out that, well, they're not really isolating it here, but it looks like a virus because it replicates RNA and it kills the cells, and it's the same RNA that's found in the person. So anyway, and so I don't want to go into too much detail in this paper because this is the pre-alpha one, and their second paper they actually compare alpha and delta. So I told you what they found in the abstract, but I want to skip ahead to the third paper. And I'm just going to quickly go through comments and make sure no one has problems with my sound or pictures, and make sure that no one's asking any questions of clarification on these studies that we are looking at so far. Doobie, a dude says, you have a great voice for singing about the juice. And that's because not using the word juice, I used the real word. I was just testing whether singing it, like I got juiced or something like that affects whether the you get the warning that pops up or not. But anyway, thank you. What was your name? Doobie? Thank you, Doobie. All right, real quick, I'm going to make sure that no one on YouTube is complaining about the screen share. All right, we're good. Okay, so I'm going to go on to the third paper. And I'm going to make, and this is the most important paper because it directly compares alpha and delta. So I'm going to go to chrismasterjohn.phd.com/juice. Are we at 3 or 4? I think we might be at 4 now. Juice 4. Is that right? No, we got 2 left. We're at 3. Okay. Juice 3, chrismasterjohnphd.com/juice3 brings up the correct paper. And so these are the same people as before. So real briefly, they're all from Johns Hopkins or NIAID. And the funding, I think the funding is from the exact same place. So it was provided by, same Johns Hopkins, NIH, Department of Health and Human Services, NIH. So it looks like there's more grants here. So it's all Johns Hopkins, NIAID, NIH, CDC. That's who's funding it. So these are all coming straight out of the major public health folks. So this is not someone in my Instagram last night wanted to know if these were all on a right-wing nutcase site. So I don't think PubMed and NIAID, CDC, and Johns Hopkins are right-wing think tanks. I don't think they are. But you never know, because I wouldn't be surprised if CNN got hold of the video I'm making now and said that I was a far-right juice opponent or something like that. So anyway, let's take a look at this paper. So this is the critical paper. Let's look at the abstract first. I'm going to spend more time on this paper because it's so important in making the comparison. So they say the emerging Delta variant quickly displaced Alpha, and it's associated with increased cases nationally. Delta has been associated with greater transmissibility and higher viral RNA loads in both juiced and unjuiced and fully juiced individuals. Data is lacking regarding the infectious virus load in Delta-infected individuals and how that compares to individuals infected with other lineages. So they whole genome sequenced 2,785 clinical isolates that were used to characterize the prevalence of the virus circulating in the national capital region. So this was DC, Virginia, and Maryland, in the areas around the capital. They might have had some. Did they have some Delaware? I forget. We'll see in a minute. National Capital Region between January and July 2021. They looked at the reviews of clinical charts to see their symptomology for the Delta and Alpha and pre-Alpha lineages, so that they could compare them between lineages across time and between juiced and unjuiced individuals. And then they did the cell culture thing that I was describing to you before. So the results are the Delta displaced Alpha to constitute 88.2% of circulating lineages by July 2021. Delta was associated with increased breakthrough in fully juiced individuals that were mostly symptomatic when compared to Alpha. Although it's important to note that when you look at the Delta people, they had been juiced earlier, because during the rollout, if it was rolled out in Alpha, then all the early juiced people were more likely to get Alpha than they were in Delta. So it's not that there weren't newly juiced people during Delta. It's just that the people who were juiced first were more likely to catch Alpha than to wind up catching Delta, because they were juiced during Alpha. So there's newly juiced and old juiced people during Delta, but compared to the Alpha, it's just on average, the time that has elapsed since people were juiced on average is greater during Delta. So the impact of juice on reducing the recovery of infectious virus from clinical samples was observed with Alpha, was only observed with Alpha. So I'll show you the data, but notice only observed with Alpha. That means that there was no effect of the juice observed on Delta at all. Okay, so anyone who comes back and says, well, no one said the juice was 100% effective. No juice is 100% effective. We're not talking about not 100% effective. We're talking about 0% effective. All right, we're talking about 0% effective. Okay. Now, someone could say, well, there might be zero difference in the likelihood of getting infectious virus, but what about the likelihood that they got it in the first place? What about the likelihood they wound up in the study? This study is not designed to look at that. If you look at that, what you see is some minor effect that has declined 80% since Alpha. And what I will also show you is that Delta was not predominant for very long during this study. And so we should fully expect that, given a couple more months, that small effect on reducing the probability that you'd wind up sampled at all, which we can glean from this in an imperfect way, seems to be there. That might not exist come 2 or 3 months, come October, what are we at? 18th, since the cutoff data for this was in July. Okay, so let's take a look at a couple things. So the first thing I want to look at is figure 1B, where you can see the red line. If you're using chrismasterjohnphd.com/juice3, or if you're on YouTube, you can see the screen share. The red line shows that Delta was basically nowhere until in May, when it shot through the roof, and that corresponded to a dropping off a cliff of Alpha. All right. And remember, their previous study didn't really show Alpha was transitioning to Delta, but they showed proof of principle that the juice was selecting for certain mutations that evade the immunity of the juice. And so it's pretty reasonable to say that, especially when we combine it with the evidence from this paper that I'll show you shortly, that seems to suggest there's zero efficacy against transmission of Delta. Basic logic says that if Alpha turned into Delta during the rollout of the juice, and if the juice protects against transmission of Alpha but not Delta, then the rollout of the juice is what selected for Delta. But anyway, So one thing to notice that the cutoff was in July and it reached a peak of 88% or whatever that number was in July. That's only 2 months looking at the transition to a Delta dominant environment. So we don't even have 2 months of a Delta predominant environment to look at. And I think the trajectory shows massive dropping off of efficacy against being sampled. That almost disappears by the study and probably would fully disappear. And then if you're sampled, there's no difference whatsoever in the infectious virus that gets cultured, so to speak, from your snot. Okay, so the next thing I want to look at is the alpha was associated with a significant. No, I want to skip over that. The symptoms is interesting, but it's not the point of this video, and I want to make this no longer than it has to be. Okay, so all right, recovery of virus in Delta versus Alpha. To assess the recovery of infectious virus from Delta versus Alpha, samples from a total of 141 alpha, 95 unjuiced, 46 juiced, and 90 delta, 63 unjuiced, 27 juiced, were used to inoculate the cells. Significantly more specimens with delta had infectious virus compared to specimens containing alpha. So just if you had delta and you were sampled, you were more likely to have infectious virus in your snot. And so that right there tells you that they talk about delta being more transmissive. That's what they're talking about, that if you got tested positive, you're more likely to find the infectious virus in the snot if you're delta. You're twice as likely. And so that's what we're really looking at. We're not looking at the probability of winding up being in a sample. I mean, that's a secondary concern. But when they talk about delta being more transmissive, this is what seems to drive it, that if you have alpha and you test positive and you sample and you have enough sample to be whole genome sequenced and it shows your alpha, you're still only 30% likely to have culturable virus from your snot. Whereas you're 2/3 likely, 67% likely if you have delta, right? So these are all people who tested positive. They're all people who had a low enough cycle threshold and a high enough amount of viral material in their nose to whole genome sequence it, but they're still twice as likely with delta than alpha to have infectious virus in their snot. That's what's driving the transmissibility, and that's the key metric, and that's the one that has no difference between juiced and unjuiced people. So I'll show you that in a second. So significant increase in the recovery of infectious virus from specimens with delta as compared to alpha was found for both unjuiced and juiced. Oh yeah, okay. No, here's the key. This is the key piece of data right here. Specimens from the fully juiced alpha group showed significant reduction in the recovery of infectious virus as compared to the unjuiced. Alpha group had 17.4% in fully juiced and 37.9% in unjuiced. All right. So that's a significant difference. It's a minority of people in both cases, but it is higher in the unjuiced people. It's clearly higher, 38% versus 17.4%. All right. Which was not the case. Listen to these numbers very carefully. Very, 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 very carefully. Which was not the case in the delta groups, which had nearly equivalent specimens of infectious virus. And this is not statistically significant, but listen to these numbers. The fully juiced, 70.4%. The unjuiced, 66.7%. It's not significant statistically, and it's nearly equivalent, but it is numerically higher in the in the juiced group, all right, numerically higher, even though it's not statistically significant and even though it's nearly equivalent. So we're not talking about this isn't 100% effective. We're talking about the efficacy is negative. It's less than 0% as a point estimate, but it's too close to call it. It's not statistically significant. But there's more of it, numerically more of it in the juiced individual. So you can't say it's not 100% effective. No juice is 100% effective. It's negative efficacy. It's not statistically significantly different from zero efficacy, but it's negative. All right. But that's not even the worst part about it. Just look at the numbers in general. Let's say that you test positive, you have enough virus in order to generate a whole genome sequencing, right? So everyone who had a whole genome sequencing probably had a CT value lower than 28, because that's generally the cutoff for are we able to sequence the genome, right? So these are all people with strongly positive tests who have enough material to whole genome sequence, and yet juiced was 17.4% had infectious virus in alpha. Unjuiced was 37.9% had infectious virus during alpha. Both of those numbers are way lower than either of the numbers for juiced or unjuiced people during delta, right? So the juiced people during delta are 70.4%. The unjuiced people during alpha were 37.9%. So if juicing drove the delta variant to take over, the delta variant is now so much more transmissible than alpha that the fully juiced people under delta are more transmissible than the completely unjuiced people were during alpha. So this is worse than, I don't know if it's worse than delayed onset gasoline being used to put out a fire, but it's not any better than that. I mean, yes, this is worse than it. So this is like delayed onset gasoline didn't just blow up the house that you put the fire out with, but it blew up the houses next to it, right? Because the effect of replacing alpha with delta is making the fully juiced people during delta more transmissible than the unjuiced people in alpha, right? It's just everyone is more transmissible now, and there's no difference. That was the 3-month effect of rolling out the juice. Delayed onset, they saw the house on fire. They said, quick, we need the best thing possible. New for-profit company says, I got the best thing possible. Delayed onset gasoline puts out a fire 95% quicker than water. We showed it in our pre-fire studies. We want you to test it out, roll it out in this neighborhood. Boom, 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 puts out the fire. Houses all around blow up 6 hours later. That's this effect, right? Okay. So now someone would naturally say, okay, well, these are the people who are sampled, but what about whether they wound up in the study at all? And that is an important point, but I want to emphasize this is not the most important point, because remember, just winding up in the study really wasn't that important back during alpha, because back during alpha, even the almost 2/3 majority of the unjuiced people did not have infectious virus if they tested positive for alpha, and they tested so positive that their CT value was so low that it was eligible for successful whole genome sequencing, right? So just testing positive and winding up in the study is not a good metric of transmissibility, because for juiced people during alpha, it was 17.4% of them wound up having infectious virus, and unjuiced people is still only 37.9%. That's the majority, and it's almost 2/3 of unjuiced people during alpha did not have transmissible virus that they could prove during the culture study. So just winding up in the study is not the key metric to look at. What we just looked at is the key metric, but to satisfy the people who want to know that, because it is a good question. we got to go down to the table. Let me I read this in the PDF and it's a little different order here. So I think here the table is let's see here. So it's not table 1. Table 2 is all clinical. There's another table here. Maybe it was in a supplement. All right, I'm going to go back to my notes. I'm going to stop sharing on YouTube for a second. I'm going to go to my personal notes on this. All right, so oh, okay. I'm sorry. It's from the Figure 2 legend. So let me go back to my screen share for YouTube folks. And so if you're looking on at home, we're looking at the Figure 2 legend. And so in the Figure 2 legend, we see the numbers of people that made it into this study. And so there were, for pre-alpha, there were 254 people. For alpha, there were 562, and for delta, there were 98. But when you separate that between juiced and unjuiced, we're looking at in Figure 2D legend, alpha had unjuiced 442, fully juiced 42. That's tenfold efficacy. It's a tenfold difference in the, and this study is not designed to look at the rate of people from a sample who test positive. That's not showing this, but you can glean as a general idea how likely they were to have samples just based on the total number of samples. And so this is a very imperfect way of looking at this, but what it shows is that 442 unjuiced alpha people made it into the study. 42 fully juiced alpha people made it into the study. So 442 divided by 42, that's basically a tenfold difference. It's almost a tenfold or a little more than a tenfold difference. Now for delta, it was 47 versus 23 people made it into the study. And so that's a twofold difference. So if you go to from a tenfold difference to a twofold difference, that's an 80% loss of efficacy over a couple of months in just the probability of them getting numbers from each group into the sample. And so again, those numbers are not designed to look at the rate of winding up in the study from the population who is juiced and unjuiced. So I wouldn't read too much into them. But because I know some people are going to respond and say, okay, well, once you're in the study, you have no difference in infectious virus. But what about the probability you got into the study because the juice is helping fewer people get sick? If you just take the numbers that are there, the imperfect numbers that are there, it seems like the efficacy against having the samples wind up in the study was 80% lost over a couple of months as the alpha transitioned into delta. And so the reason that tells you that we should infer nothing from this is if we go back to Figure 1B, we see that delta started taking over in May. It wound up taking over by July, and July was the data cutoff. So we don't even have 1 month of this being, in June, delta was less than 50% of the cases. So we don't even have a full month of delta predominance here to allow the 80% loss of efficacy to turn into total loss of efficacy. I mean, it doesn't take, if that is an 80% loss, it doesn't take much extrapolation to say that might be 0% efficacy or negative efficacy once you spend 2 or 3 months in a delta predominant state. And then, okay, so overall, what did we show here? So we showed that the juice protected against the So first of all, we showed that during alpha, before delta took over, if you tested positive and you had a low enough CT value, meaning you had a strong enough test that they could do whole genome sequencing and verify the variant, both juiced and unjuiced people probably did not have infectious virus in their snot. Okay, 17% of juiced, 30-something% of unjuiced. There was more than a 3/4 likelihood overall and a 2/3 likelihood, even for unjuiced people, that they did not have infectious virus. So during alpha, just most people weren't transmissible, even if they tested positive. But there was a transmission reducing effect of the juice. The transmitting effect of the juice seems to have selected for delta, because if the juice protects against transmission of alpha but not delta, and it stops transmission of alpha, then that makes room for delta. And it can't reduce the transmission of delta, and so that provides all the focus on the juice as the one thing, therefore leaves the population totally vulnerable to the takeover of delta, because that population believes the juice was the only thing they needed to protect, and the juice does nothing to protect against the transmission of delta. So that's delayed onset gasoline. But the takeover of delta was worse than that makes it sound, because it not only led to the loss of juice efficacy, but it made everyone juiced or unjuiced more transmissible, right? Everyone more transmissible because the juice selected for delta. So that's delayed onset gasoline that put out the fire very quickly, but didn't only blow up that house 3 hours later, but it blew up the rest of the houses on the block. All right. Just for the sake of my lighting, the sun has moved in a way that hold on. All right. I don't know if that's better, but I think that's better. Okay. All right. So now we're going to go to, and by the way, I will, at the end of this presentation, I'll be fielding any questions in the live chat. Solemn Sugary Soy Sludge says you need to post a short of this minute with the blowing up the houses part. Oh, I fully intend to do that. Just making sure no one's complaining about my sound or my video. Okay. So yeah, I mean, I'm going to make a short out of this. So that, for the people that don't want to sit here for an hour and a half looking at 5 studies. All right. So that was chrismasterjohnphd.com/juice3. And now we're going to look at chrismasterjohnphd.com/juice4. And so if you're on YouTube, you see the screen share. And if you're elsewhere, you can just type in, if you want to follow along, chrismasterjohnphd.com/juice4, and you'll see the study that we're talking about. I want to make a point here. So this paper is from Morbidity and Mortality Weekly. All these other papers were preprints, which means they're currently being peer-reviewed. So for the not peer-reviewed trolls, every single one of whom has never, ever published a peer-reviewed paper and doesn't know anything about the peer review process and has never peer-reviewed a paper, those are the only people running around going, but it's not peer-reviewed, and on the preprint site, it has a disclaimer. I want to make it clear that nothing that occurs in MMWR Morbidity and Mortality Weekly reports by CDC, which is indexed for PubMed, none of that is ever peer-reviewed. And so just to emphasize this point, let's just look at what CDC says. So we're going to Google, is MMWR peer-reviewed? And let's look at what the CDC tells us about their journal. They say, although most articles that appear in MMWR are not quote unquote peer-reviewed, they use quotes, in the way that submissions to medical journals are, to ensure that the content of MMWR comports with CDC policy. Every submission to MMWR undergoes a rigorous multi-level clearance process before publication. Okay, first of all, no scientific journal that's peer reviews to make sure that it comports with anyone's policy. That's not what peer review is. Peer review is to make sure that there aren't mistakes, errors, and gross problems with the logic in the paper from people that study that thing, that know, like, did they use the wrong method to measure this? Did they misinterpret it? That's what peer review is for. And every journal, whether it's scientific or not, has an editor who determines whether the publication of that thing would comport with the policy of the journal. That's called having an editor. That's not called peer review. And if they're talking about CDC as a government agency, if they're talking about their policy, not for the journal, I'm not sure what they mean. But if that's what they mean, then no one in their right mind would consider it even remotely analogous to peer review for someone to make sure that the article comports with a government bureaucracy's public policy. That's sort of, science is not comporting with policy. Science is rigorously analyzing stuff. So I just want to make the point that none of these papers are peer reviewed, but it's only the CDC paper is the one that will never be peer reviewed, because the CDC MMWR is not a peer reviewed journal. And it's funny that the CDC uses quotes when they say MMWR are not peer reviewed, in quotes, in the way that the peer reviewed literature is, as if that's not important. As if making sure that comporting with CDC policy was more or less the same thing that they do at the other journals, where a collection of researchers from different countries and not who don't work for the journal evaluate the science based on its merits. Okay. Anyway. Okay. So this is during Delta. So let's look at what happened in this prison during Delta. So from the abstract, actually, let's go to the full text and see if there's any funding information and where the authors are from. So the authors are, oh, what happened? All right, the authors are from CDC. All right, guys, my Instagram crashed, so I'm just going to go live again real quick. All right, we're back on Instagram. Okay. So these authors are from CDC and from the U.S. Department of Justice Bureau of Prisons. So this is from the government, and let's see if there's any funding listed. Funding doesn't come up in a keyword search. Support sometimes is, no, that's PubMed support. That's coming up. PubMed Support Center. That's not what I'm looking for. What other words do they use? It looks like they don't cover, oh, in the acknowledgement sometimes they do it. No potential conflicts of interest were disclosed, but it doesn't seem to list the funding anywhere in this. Okay. So let's go back to the abstract just to look at an overview what they're finding. Incarcerated populations have experienced disproportionately higher rates of COVID-19 related illness and death compared with the general U.S. population, due in part to congregate living environments that can facilitate rapid transmission the virus and the high prevalence of underlying medical conditions associated with severe virus. The Delta variant has caused outbreaks among juiced and unjuiced persons in congregate settings and large public gatherings. During July 2021, the virus had an outbreak involving the Delta in a federal prison in Texas, infecting 74% of incarcerated persons in 2 housing units. The Federal Bureau of Prisons partnered with CDC to investigate. CDC analyzed data on infection status, symptom onset date, hospitalizations, and deaths among incarcerated persons. The attack rate, meaning who got sick, the attack rate was higher among, and now when we look at the data, we'll see some problems with the summary. Okay, but I'm just reading the abstract. So the attack rate in this Delta dominant outbreak in a jail, the attack rate was higher among unjuiced versus fully juiced persons. Notice how high these attack rates are. And notice how low the difference is because almost everyone got sick. So 93% of unjuiced people and 70% of juiced people in the housing unit got infected. That's, almost everyone, like so many people got infected that there's a difference between those two values that is statistically significant, but you can't call that 95% efficacy. You can't call that very very effective. So I mean, just to do that real quickly. So 70 versus just the raw data, 70 versus 93 is 75% as many people who were juiced got. I mean, I don't know if this is exactly how they calculate the efficacy usually, but I would call that 25% fewer people got infected if they were juiced. And I'll show you why there are some concerns about that summary, but that's showing really low efficacy just against getting infected. Now, granted, it's close living in a prison, and so that's why the risk of getting infected is so high. But it's also interesting that hardly anyone got very sick during this as well. That's also interesting. I mean, but it just goes to show you that the higher the risk, the lower the efficacy estimate. So it's like it's really effective when it doesn't matter, but it's not very effective when it does. Okay, so among a subset of 70 persons consenting to an embedded serial swabbing protocol, the median interval between symptom onset and last positive test, so how quickly did they get viral negative, in fully juiced versus unjuiced persons was similar, 9 versus 11 days. It wasn't statistically significant. So basically, if you got sick, you didn't get better any faster if you were juiced. And you had 25% efficacy against getting sick if you were juiced. But and I'll show you why that's problematic in a second, but that's the raw overall data. One or more specimens were culture positive from 42% of juiced and 38% of, 42% of unjuiced and 38% of fully juiced persons for whom bioculture was attempted. So it's slightly higher, but not statistically significant for the unjuiced people, right? So this is numerically higher for the unjuiced people, but it's not statistically significant. The other study we looked at was numerically higher for the juiced people, but it wasn't statistically significant. So maybe you can pull these studies together and just say it makes zero difference, zero efficacy against culturable virus and transmissibility. Okay, so on the surface, what this shows is that there's a very minor 25% efficacy of the juice in getting infected. But if you were infected, if you tested positive, were you transmissible? So there's no evidence that there's any difference in transmissibility between juiced and unjuiced. And so if you just look at the surface data, you would say as a personal choice, you're less likely to get infected slightly, but you're probably going to get infected if you're in a prison no matter what. But you're no more or less likely to transmit it. And now, but remember, it's the transmissibility once you're infected that matters, not how many people got infected. Because remember, like back during Alpha, for example, if the juice is making Delta take over from Alpha, remember back during Alpha, most people weren't transmissible. The majority of people weren't transmissible regardless of juice status. And so the overall effect is clearly on this lack of difference of transmissibility and shifting over to the Delta variant that's more transmissible in everyone. But I want to go to the full text of this just to show you one of the problems with looking at the summary data, and that is the different juices. So if we look at the attack rate For unjuiced, it's 93. For partially juiced, it's 67. For fully juiced, it's 70. It's interesting right there that fully and partial is not different. But what's nuts, and this, if you're following along, this is in table 2 of the study. So if they separated it by the type of juice, Mr. Johnson had 77% efficacy. Excuse me, no, not 77% efficacy. 77% of people got infected with Johnson. For the modern juice, you know what I mean, it was only 40%. And so for the fuzzy juice, I think you know what I mean, it was 81%. Now, part of the reason that it might have been, now, first of all, 81%, 93% are hardly any different, right? But part of the reason that, so, and then second of all, that 25% efficacy is basically completely from the modern juice and not from Mr. Johnson juice or from fuzzy juice. But the majority of the prisoners got the fuzzy juice, and the fuzzy juice, which has been used in Israel and is known to have very poor durability, was on average, most people had gotten the fuzzy juice 4 months ago. And so they are brought to almost zero efficacy of the fuzzy juice compared to unjuiced. Like 81 is a little bit lower than 93%, but like it's not that much different, right? And so after most people got it 4 months ago, the efficacy is almost gone. And then that, so, just to calculate that efficacy, and I'm not going to do stats on it, but just to get an eyeball of the effect size, 81 divided by 93 is 87%. So that's 13% efficacy of the fuzzy juice after 4 months. So after 6 months, what was the efficacy of the fuzzy juice going to be? Probably nothing. But still, that's, very modest efficacy of the modern juice and Mr. Johnson juice, close to 0% efficacy of the Pfizer juice against getting infected, and zero efficacy against transmissibility. Now, it's also interesting. This study is just interesting that these people are in very close quarters. They have very high infection rate, and presumably they would have high viral loads if they are in such close quarters and have such spread. But let me just, somewhere in here they have, where do they? So somewhere in here they have the number of people who went to ICU and died. Let me just keyword search died. Oh, not ICU. So four people were hospitalized, 3 were unjuiced, and 1 died. It's rather phenomenal that so out of 223 people, 172 got infected. And so the case fatality rate was, 1 second. The case fatality rate was 0.005% in very close quarters, where Delta was so transmissible that 3/4 of the people got infected, all living in close quarters to make it very transmissible. And it's 0.5% case fatality ratio. That's interesting. And prisoners are probably not the healthiest people around. So anyway, this is consistent with in Delta, very tiny effect that is wearing off very quickly from previous estimates against getting infected, very tiny effect, and no efficacy against transmission. All right, so the last one we're going to go to is chrismasterjohn.com/juice5. And I've done a video on this previously, but this answers a question. Like, say that this is all sampling bias, and it's just, once you get infected, that's when you're equally transmissible, but juice people aren't going to get infected. And so there is a problem with that. But as I showed you, the numbers derived from these papers don't really support that except as a slight effect. So we saw that in the national capital region, that the efficacy against not true efficacy, because it wasn't designed to calculate that, but just based on the number of samples they had, juiced unjuiced difference was tenfold for alpha and was twofold for delta, indicating 80% apparent loss of efficacy over that time period. But they weren't even in delta dominant state for a month. And so that 80% loss of efficacy could probably be a total loss of efficacy by now in October. That cutoff was in July. And then in the prison population, we saw that overall there was I forgot what I said. Was it a 20? It was somewhere around 25% efficacy. I forgot what the exact number. But for fuzzy vaccine, damn it, I'm not getting the code words right. So for the fuzzy juice, it just in a few months it dropped to 13% efficacy, right? And so the general trajectory is total loss of fuzzy vaccine in even against getting infected at all. But the reason I want to look at this last paper is because they looked at a subsample of asymptomatics. And so presumably the asymptomatics, presumably they were not subject to the same degree of sample biasing where because they were sick, they got tested. Because these are people who got tested for some other reason than they got sick because they weren't sick, right? So the asymptomatics should not be subject to this sampling bias problem. Now, granted, the asymptomatics are a small percentage of the population in this study, and so we don't have big numbers, but they're very important because across the 5 studies I've found so far, which is, I believe, all that are indexed for PubMed plus some, all preprints, and if there's anything else, I believe it was probably published very recently and in the preprint servers and not indexed for PubMed yet. But I did go beyond PubMed for this. So in the 5 I've been able to find so far that cultured infectious virus from snot, this is the only one that looked that had a sizable sample of asymptomatics. Okay, so Chris Masterjohn, phd.com/juice5, or just follow the screen if you're on YouTube. And let's look at the authors and the funding. So the authors are all from University of they're from University of Wisconsin-Madison. They're from Public Health Madison in Duane County. So the local public health department for Madison, or they're from CDC Atlanta. All right. So we're talking Public Health Madison, University of Wisconsin-Madison, and CDC Atlanta authors, right? Not a right-wing think tank. All right. Not a right-wing think tank. Okay. And then the funding. I can't even believe that I'm pointing out that this is not a right-wing think tank, but I literally had someone ask me on Instagram yesterday if these studies are all published on a right-wing nut job site. All right. So the authors declare no competing interest. The work was supported by CDC contracts and DHO and TCF. What's DHO? What is DHO? I don't know what DHO and TCF is, but it sounds like some government agency. Anyway, funding came at least from CDC. Authors from CDC Atlanta, Public Health Madison, University of Wisconsin-Madison. Okay. So let's go to the full text. And so what they did was they looked at CT counts under 25, which in previous data, and I've been extracting all the CT counts with different tests to match them for the probability of getting infectious virus. And I totally agree with them that all studies should be below 25 as a cutoff if they want a high probability of culturing virus, no matter what gene they're measuring. But anyway, so in figure 1A, they're showing that the distribution of CT counts across all cases, unjuiced and juiced, looks exactly the same. And if you take a cutoff above 25, which would be around here, it also looks exactly the same. So there's no difference in the probability of having a CT count under 25. So then the second thing that they did in figure 1, well, no, the second thing is figure 1B. Well, actually, I want to go to 1C first. So in figure 1C, what they showed was in a subset of juiced and unjuiced samples with CT below 25, they looked at plaque-forming units in culture and found that there was no difference between the two of them. So just same distribution of the amount of virus cultured. Then they have, I want to skip ahead to supplementary figure 2, shows whether infectious virus was cultured or not. And in the juiced and unjuiced with CT values under 25. And so what they showed was almost everyone in both cases, with no difference between the two groups, juiced or unjuiced, had infectious virus cultured when it was under CT 25. So the other results are not using culture from infectious virus, but because they showed that CT under 25 in a subsample was indicating in their test, was indicating a very high probability of infectious virus with no distribution in the CT values between juiced and unjuiced, this gives us a pretty reasonable belief that just looking at CT under 25 in the larger sample does truly reflect some 90-ish% chance of infectious virus. Okay. And so it's unfortunate that they could only do it in a subset, but because they validated it in a subset and showed that it's no different, the relationship between CT count and infectious virus, no different in presence of infectious virus between juiced and unjuiced, I then give more credence to the CT under 25 values in the rest of the study because they validated it against the viral culture. That's why I'm not including in this other studies that did show differences in PCR positivity that didn't even try to look at culturable virus. So now if we go back to figure 1, what we see in figure 1B is where this super important observation is. So in symptomatics, no difference. In unknown symptom status, no difference. But in asymptomatics, they had a 30% greater likelihood of having infectious virus in their snot, indicated by CT values under 25, verified in the subsampling that I just showed you. The juiced P equals 0.05, so borderline, right on the border of conventional statistical significance, had were more likely to have CT under 25, indicating infectious virus, than unjuiced. And so it's a very small sample size, and it's not a random sample of the population, but this is the only paper that cultured infectious virus that looks specifically at asymptomatics. And so what that's showing is that the random person on the street who doesn't know who they're sick, that's the person who's not staying home from school, who's not staying home from Broadway, who's not staying home from the restaurant. That's the person who sees no reason to not shake your hand. That's the person who sees no reason not to go up to the 75-year-old person and speak face-to-face to them within spitting distance. That's the person who, for all intents and purposes, has full reason to believe that they're not sick. That's the person across all these 5 studies. That's the profile of the person that is more likely when juiced to have Delta virus culturable from their snot than unjuiced. So let's synthesize this. During alpha, juiced reduced transmission. Because juice doesn't do anything to Delta, and it does reduce transmission of alpha, juice causes alpha to be restrained and says, make room for Delta, and says, Delta, you have free roam in this population. Then juice then the government, on behalf of juice, says, there is only one thing that you need to do, and that's the juice. And so government then tells the people, or some governments like New York City say, if you're juiced, you are safe as long as we fire every last unjuiced person. If you're juiced, you are safe to go to Broadway as long as we don't let any juiced people inside. If you're juiced, you're safe to eat indoors at the restaurant as long as we don't let any unjuiced people in. So this then teaches the lesson that you should be afraid of unjuiced people, but if you're juiced, you can relax. You don't need to do anything to protect against transmission. And the government doesn't say this to anyone, but within the distribution of people who listen to that message, there are the people who think that they can pick their nose and then serve someone coffee at Starbucks because they heard that message. I'm not saying the government's telling people that. I'm just saying that there are different levels of conscientiousness in the population. And there is a certain type of person who hears juiced people are clean and unjuiced people are unclean, and we kicked all the juiced people out, so now you don't need to worry, who now says, well, no one saw that I picked my nose and I wiped it on my pants, and I'm not sticking my finger in their sandwich, so it doesn't matter, right? Don't tell me that person's not out there, all right? And so the net effect of the messaging, layered on top of the fact that the juice doesn't protect against transmission of Delta, says that, okay, we got rid of Alpha, we made full room for Delta. Delta came in, and then we made everyone behave and think that now they can relax. So Delta, which already was biologically more transmissive, now has everyone off their guard and just amplifies the fact that the juice does nothing to the transmission. But more to the point, the fully juiced person during Delta is more transmissive than the unjuiced person during Alpha was. All right. So the juice made Delta take over, which made juiced and unjuiced all way more transmissive. And to top it all off, across these 5 studies, the only thing that looked just at asymptomatics showed that if you're the person who thinks you're not sick and there's no need for you to be cautious around anyone, that you are at a 30% greater likelihood of having infectious virus in your snot. And unfortunately, a subset of the people are the people who hear that message and think that they can pick their nose and, as long as no one's looking, that they can then wrap up someone's sandwich. And so I go back to what I said at the beginning, and I will make a short version of this. The juice reduces transmission like delayed onset gasoline puts out a fire. And after the data cut off for your study, not only does the house blow up, but so do the rest of the houses on the block. All right, I'm done with my spiel. I'm going to go look through the comments in the live chat. I got a bunch of live chats going. In Instagram, we crashed. And so if you left any comment in the live chat previously on Instagram, I've lost it. So if you asked a question, you got to ask it again. Koz Nina says, need that shirt. I made this shirt and I paid extra from Rush Order Tees because I only bought one of them. So if people want the shirts in my videos, they got to tell me. And, maybe what I'll do is either bulk make them and sell them at cost, or bulk make them and devote and like give the proceeds to fired teachers and healthcare workers or something like that. But you got to give me feedback if you actually want the shirts, because most of the shirts that I'm wearing with the exception, there's a couple exceptions, but most of them I made them myself. Um, and then I'll have to get some kind of form to estimate how many people will buy them, because I don't want to tank a bunch of capital into shirts that people aren't going to buy. But I would definitely be happy to find some way to get them drop shipped and have like the proceeds go to a GoFundMe for people who lost their job over the mandates. Christina Marie says, what study are you looking at? I called it out as I was going along, but all 5 studies, you can go to chrismasterjohnphd.com/juice1, brings up the first study. Same thing, juice 2 brings up the second, juice 3 brings up the third, juice 4 brings up the fourth, and juice 5 brings up the fifth. Caramel Jones says, obvious lies, manipulation for goal of control. That's not an unreasonable inference. Caramel Jones says, control, control, control. Certainly looks like it. Caramel Jones says, what do you mean you crashed? I'm viewing you now on IG. So you're viewing the second live after the first one ended because of a poor connection. So you're watching the second half. All right, I'm gonna go to YouTube now for the live chat. Show me what I need to know says, great vid. Thank you. FEMA Jacob says, what is he reading? I just got here. You can go back to the beginning of the live stream, just scroll it back. You can listen at 2x speed and catch up if you want, or watch the recording when it's over. But you can also click the links in the description on YouTube to see the studies we're talking about. Johnny Coe says, just got in. What is the juiced? Look at the screen share. What I'm talking about. It's the thing that's being mandated that goes in the arm. Jan 2052 says, I'm on the fence for getting juiced. Employer concerning me will lose pay. Health insurance 401k have to decide by October 31st. I'm healthy, not worried about infection, but I worry how juice will change immune system. Well, I can't tell you what to do. I understand that people who are facing attacks on their livelihood are forced between a rock and a hard place if they're undecided. And I'm working hard to help people make the decisions they need to make on if I can't make you decide. I can't decide for you your values, right? I can't decide for you whether you should comply with the mandate to keep your job or whether you should take some other course of action. I am trying to do research, first of all, to help legal efforts to give you the freedom of choice. And then when I'm done with that, second of all, to help, if you do make one decision or the other, to help you navigate that decision with your health. But I can't decide your values for you. So I understand that that's a very difficult decision. I'm sorry that you have to be in that position, and I'm doing everything I can to make people not have to be in that position. No Agenda Show video says, how do you know that Alpha or Delta actually exist, though? Well, I mean, the RNA exists. Now, some people are questioning whether it's a virus. That'll be the last thing that I look into, but I'm pretty sure that what I'll decide when I do look at that research is that maybe they didn't perfectly isolate the virus, but it behaves like a virus. I mean, you're talking about something that, one, has exponential spread behavior like a virus would. You're talking about something, two, antivirals kill it like they would kill a virus. You're talking about something, three, things that feed viruses, in some case feed its growth. There's some studies like that. You're talking about something, four, that so there are some studies. So it's interesting that in most of the antibody studies, they used pre-pandemic serum as a control. And they never did that with any of the PCR studies. But there are a couple studies where they looked at pre-pandemic PCR tests on samples that were there to try to define the exact beginning of the pandemic. And in one case, to try to see if cats transmit it. And in 3 studies that I found, there was no PCR positivity before the pandemic. And so that it's not designed to show that, but it seems like in the short period of time before the pandemic, it wasn't showing up in flu samples. So that's a piece of evidence that it wasn't always there. But then in these viral culture studies, yeah, they're not isolating the virus and looking at every last thing with an electron microscope and stuff, but they are showing that something that the person tests positive for, that at least in the 6 months before the pandemic, no one was testing positive for based on stored samples. And I mean, that's not real. I'm not super confident in that, but what I could find suggests that. That thing at a high enough concentration kills the cells and then self-replicates. So it certainly exists, and it looks like a virus and behaves like a virus. And that's all I can say. Snowman Mike, do you think in 5 years now the juice will affect people? How does the data look so far? I mean, I don't know what happens in 5 years. We don't have any 5-year data. How do I know? Troby3D3R says, love the juice on juice term. I got that from Australia. So if you go to We the People Are the News on Instagram, scroll down and there's a 4-way convo that, well, it was one person was on and then a 3-way well, no, it was so there's one guest, then I came on, and then 2 more guests came on. And so John, the host, we did a 4-way. One person was from Australia, and they had a whole set of code words that they have to use because all our videos are being scanned for deplatforming. And so I'm using it based on experiments I did with just saying different words in Instagram and seeing when the warning pops up. I'm just guessing that the warning is a rough metric of the cumulative risk of future deplatforming. And so I don't want to be overly paranoid, but I am trying to do the minimum needed to be not confusing if I can, but still try to avoid the obvious tags for warnings, because I'm assuming that the more warnings I get, the more I'm tagged for pushing me down in the algo and eventually getting rid of me. I'm just trying to be modestly conscientious about that. Snowman Mike unfortunately got the first juice last Friday. Canada is crap, man. I mean, that's I believe people should have the freedom to get whatever juice they want. But when you take away that freedom, you have a lot of buyer's remorse among people who never wanted to buy in the first place. And so I don't think these people have any idea what kind of blowback they're getting themselves ready for, but we'll see. Solemn Sugary Soy Sludge Sobriety says you need to short of this minute with the blowing up houses part. I'm going to do that. Solemn Sugary Soy Sludge Sobriety says hit the like so people get notified by some miracle. Yeah, please like, share, and subscribe. And in the description or at chrismasterjohnphd.com/support, if you could find some way to support me by buying one of my information products as low as $2.99, half of a fancy cup of coffee, or make a donation for 50 cents or $1 million, whatever you want. Or buy one of my more expensive things, or buy something at no extra cost to you that you were going to buy anyway, but you use my affiliate links for. If you find this stuff valuable, I would appreciate it if you could do any of that. But certainly like, share, and subscribe if you like the video content, because that's what keeps the video content circulating. Sandra doesn't like juice. Rumbecker says, you do not need a study to show that juice would make super spreaders. If less likely to be symptomatic and think you are protected, juice people would just take more chances, human nature. What you're saying is true if the juice doesn't do anything to the infectiousness. But the point of my video is that I was originally asking the question over the course of my research, does it or doesn't it? And my position has been evolving, but my current position is it did. back during alpha, made Delta take over. Now it doesn't. And so it created this situation in which it has no protective effect. But that's not obvious on the face of it, because if you're less symptomatic and you have less virus in your nose, then that would make you less likely to spread it. But what these data indicate is that once Delta took over, there is no effect against the virus in your nose. I want to make one more point. The study that we should have is a random sample of the population that's very large, just vaccinate, just juice status, infectious virus in the nose, and just answer the question in a random sample. Are you effectively filtering out the people with virus in their nose with these mandates? Because if you are, you can make a utilitarian argument around you're reducing it, and then people that are anti-mandate have to go to, maybe that won't last forever on utilitarian grounds, or have to go to the moral rights and liberties argument. But if you can't show in a random sample of the population that juice status determines the probability of infectious virus in a way that filtering out the unjuiced makes people safe, then your public health policy is actually totally irrational on utilitarian grounds. So for juice's sake, do the damn study. What's going on? All right. They don't want to know the answer. I mean, that's what's going on. Martin Zero says, do you need to get on Gab so that you may express yourself like a normal human being? I mean, I could just, the issue is that I'm going on to 4 platforms simultaneously that all have different algos, and I can go on Gab and have no followers, and I can tell everyone to go on Gab, and then 4,000 out of 100,000, 150,000 people following me can go on Gab. Or I could actually reach everyone and then reach additional people. The fact of the matter is that, yes, we need to build alternative platforms, but I think, if everyone thinks all the people in the know are on Rumble and Telegram and Gab, and then just go there and don't go on YouTube, Instagram, Facebook, and Twitter, then all they're doing is creating a siloed echo chamber where they talk to themselves and reach no one. And so I think we need every strategy. Yeah, we need to build alternative platforms, but we also need to reach maximum people. And then, and I would point out that, despite getting warnings from Instagram that I might get deleted, my Instagram is still growing and the content is getting really high engagement. So I think abandoning Instagram would be nuts. I think it would be totally self-defeating at this point. But also, I really want to emphasize, I want to keep this part of the discussion. I firmly believe that any alternative free speech platform that depends on central servers is vulnerable. And they may be the present of free speech, but they're not going to be the future of free speech. And the future of free speech will be built on BitTorrent modeled technology that replaces central servers with peer-to-peer file sharing. And I think that's extremely important because we know that Telegram, for example, has been bullied by Putin to shut down the electoral activities of his opposition, which is, Telegram's free speech, but Telegram gave in to Putin a lot faster than even Google is giving in to the federal government, and Google is happily compliant. Dorsey basically gave them the finger and said, I don't think Twitter should be the arbiter of truth. Granted, I think the Twitter Safety Committee disagrees with Dorsey, but Dorsey, I think, believes that. And so Telegram's way more giving in than Twitter, but they're also more giving in than Google, because just look at the 12 people that were called out by the federal government as should be universally deplatformed. They're still not. And so I don't think that small pro free speech platforms, I think we should go there, but I don't think we can trust them more than big tech. And you saw what happened to the one that got taken down over false accusations that the thing that happened in January was mostly organized it when it was actually mostly organized on YouTube. You saw how quick they were deleted from hardware from the App Store on devices, and how AWS just took down their cloud storage. Anything depending on central servers is extremely vulnerable. But and the App Store you could get around, right? Like you can still promote something on a website, but if you can't run it because the central servers are taken away, you're vulnerable. Rum Becker says, for real, after this video today about the juice, his days are numbered. I don't know. I mean, are my days numbered because I reported what the CDC, Public Health Madison, Johns Hopkins, and NIAID said? I mean, Fauci is personally the science, right? But if the F man is the science, then NIAID should be at least a representative of the science, right? Enrique Schandorf says, funny that absolute risk reduction, according to Lancet, was only 0.84% for Mr. Fuzzy, 0.84% for Mr. Fuzzy, 1.3% for Mr. Johnson, 1.2% for the modern, and 1.3% for the Aztec juice. I totally disagree with all these people criticizing these studies for not focusing on absolute risk reduction. I think that it's totally good and normal and reasonable that relative risk reduction is the default to report, because if something's 100% effective, then all the people who would have gotten sick didn't get sick. Something's 50% effective, half the people who would have gotten sick got sick. That's very intuitive to understand. And that is not at all intuitive to understand by just citing absolute risk reduction of 1.2%. You can't even conceive of what that means unless you also state what the absolute incidence was and what the final absolute risk was. Like, you need way more information to make heads or tails out of what that means, and you have to do math with it. But not only that, but yes, it's misleading if you state the relative risk and fail to point out whether the risk is common or rare. But it's also misleading to state the absolute risk reduction and not state the duration of the trial and point out how that would change over a longer period of time. So if 5% of people per year get sick with something, then the total cumulative incidence of the sickness is going to be 5% at 1 year. It's going to be 2.5% at 6 months, 5% at 1 year, 10% at 2 years, 15% at 3 years, 20% at 4 years. And your absolute risk reduction is cumulatively is going to go up and up and up and up and up with time. And so if you just take a 6-month study and state absolute risk reduction, you're not taking that into account at all. So yes, you do want to state the absolute risk so that you can put into context whether the relative risk reduction was in something that is common or rare. But absolute risk reduction is not at all superior to relative risk reduction as the default reporting of the effect size of something. And it is nowhere near as intuitive to understand as relative risk. Defo Radio says, I hope your videos are backed up. Well, they're not because I'm recording them live, but they will be on 4 places. And I really doubt, not 4 places, 3 places. But I really doubt 3 platforms are going to deplatform me before I can download 1 video. I mean, this video isn't any more or less scientific than any other videos that I've made that haven't gotten taken down. The only video that YouTube has taken down was one I did on zinc dosing. And I did a lot of videos on the C word. Will says, fantastic presentation. Thank you. Will says, I'll archive the video. If anyone else would like to as well, use YouTube-DL for educational purposes. Cool. I mean, I totally support like people screen recording anything that's controversial or otherwise capturing it and distributing it, because that would mimic the effect of distributed servers. Just by, if you just share from multiple platforms of video that you expect to disappear, it won't be able to disappear if there's so many different servers hosting it and so many different places it's shared. Rotha says, thanks for what you've been doing lately. You're appreciated. Thank you for the appreciation. I appreciate that appreciation. Duke Man doesn't drink juices, doesn't make juices, take juices, or inject juices. I support your right, Duke. Tommy Sandlier says, can you please explain how to find the data from England showing higher juice population hospitals? I know I saw you or someone filter for it, but I can't find it on the government site. Yeah, I'll show you that right now. So for YouTubers, I'm going to share my screen. For Facebook and Instagram, so YouTube should be looking at this. Just follow along with what I'm saying. So if you Google PHE technical briefing, top result is investigation of variants. You click on it. Then it gives you technical briefing 23 is the most recent technical briefing. You click on it, you download it, you open it up, and you could keyword search death. And look for the table that has deaths within 28 days of positive specimen. That in this case, that's table 5. I mean, if you look at these a lot, you could just scroll through it and you know where it is. But it's the bottom row in table 5 in this case, and that tells you deaths in juiced and unjuiced. I like to exclude partial juicing from the analysis because the immunity is in an unclear stage. And so I like to just look at post 14 days, post second juice versus unjuiced. And actually now it's a slight majority of deaths are in unjuiced. But if you look at technical briefing 22, you can see that wasn't the case just weeks ago. And so it's dynamic data that's changing. But for many months, it was majority juiced that we're getting it. And that also seems to be turning in Israel from what I looked at last, where the third juices are starting to turn things favoring more protection. That obviously is short-lived. Duke Man says, no one who's sane is anti-juice. They just want safe juice. Well, I respect people's right to be anti-juice, but I don't respect anti-juice being used as a dismissive term by the media, creating a narrative through which people who are brainwashed hear it and then call anything critical of the narrative anti-juice. I mean, that is brainwashing straight up. And so if you use that word in that way, you are a brainwashing victim and you should get help by not watching the TV. But I respect people's right to be anti-juice if they have some cogent argument against juice in principle. That's not me, but I respect people's right to be that. I wouldn't say they're insane. I don't know if I agree with them, but that doesn't make them insane. I mean, although some people, may not have a good argument, and they might just believe in some kind of, cult-like fealty to the idea of being anti-juice, and I wouldn't call that like a logical argument. But some people have logical arguments about people are generally overjuiced, or, if they have a logical argument, and more power to them. I think logical arguments should be refuted, not dismissed because we call people crazy. Fabian Popa says, great, Chris. Maybe state your position at the state of these. As I understand, you're not anti-juice, just for better juice, nutrition, freedom. There's no misunderstanding. Well, right. But I did state my position at the beginning, which had nothing to do with my values. And my position was on the interpretation of the scientific data. And that is the way I described it, that the juice reduces transmission like putting out a fire with delayed onset gasoline. So Arter Wisniewski says, Chris, we follow to Odyssey or else not to worry. I am not bullish on Odyssey. So I like that Odyssey is based on LBRY network, which is peer-to-peer file sharing, which is based on BitTorrent structure that avoids central servers. But there's two things I don't like about Odyssey. First of all, they did an ICO, initial coin offering, with their coin, and they're in a battle with the SEC that they're not going to win, and they're not being strategic about trying to win it. They're deluded in how they think they're going to win it, and they're not. And that coin has lost all its value. And although I wouldn't be there to monetize. I think if their coin, through which people in the Odyssey creator network monetize their videos, has no value and is destined to have no value based on how they're interacting with the SEC, then I think that the creator network's not going to stick around there, and it's not going to be a good place for discovery in the future if the creators can't effectively monetize. And then second of all, do a YouTube search for Ludkey Odyssey. Actually, if you just do a YouTube search for Odyssey, it's one of the #1 videos. And he goes on to explain how their corporate direction is extremely misguided in promoting totally raunchy, meritless vulgarity at the expense of everything else, and how they were extremely rude to him as a major, top Odyssey creator. They were who had their ear. They were extremely rude to him and vulgar to him personally when he tried to explain why he thought the corporate direction was misdirected. And so that also gives me no confidence in that the corporate direction of Odyssey on two fronts means Odyssey has any future of a vibrant creator network. So although I agree with the technology and I like the core technology, I'm very pessimistic about the future of Odyssey's creator network. Google Spy says, I'm not going to listen to people that kill the most people on what to do with my body. It's not happening. It's that simple too. Well, I'm not making any accusations like that, but I respect your right to consider it that way. I'm just, due to the length of this video, the fact that I have a lot of work to do, and the number of questions, I'm only fielding questions on the current topic. Will points out that YouTube has the market share and best clients. It's tough. I agree. Will says, imagine a social media platform that's governed by the Constitution. Oh, wait. So I think constitutionally, big tech has the right to censor people, but they're not censoring people as private companies. They are clearly and indisputably censoring people in response to continued threats of the federal government to engage in antitrust against them or to Section 230 them and change their liability for their content if they don't censor the specific things and specific people that the federal government is explicitly calling them out to censor. And the explicit people who the federal government is explicitly calling them out to deplatform, while 4 times over 2020 bringing their CEOs in to testify and spending hours during that session accusing them, throwing accusations against them. Meanwhile, the threat of antitrust action and Section 230 rearrangement lies behind that. They're not even thinly veiled threats. They're unveiled threats against them. And that has been decided by case law to be a First Amendment violation on the part of the federal government. The federal government is systematically in violation of the First Amendment. Psaki is in violation of the First Amendment. Everyone in the White House, I forgot what committee they have. The administration made a committee to look for misinformation. That committee is in full violation of the First Amendment. And all the senators that call out all the tech CEOs to censor people are in full violation of the First Amendment, and lawsuits need to be waged against them, and they haven't yet. But I fully believe that if that gets to the Supreme Court, they will all be found in violation of the First Amendment. And if big tech deplatforms someone and the federal government had threatened them and told them to deplatform that person should be suing the federal government, and that should be, deplatforming is prior restraint on steroids. Do a Wikipedia search for prior restraint and look at the Supreme Court law on prior restraint. Deplatforming is someone is saying, we, the government, determined that this person Not only can this person not say things that we reviewed prior to allowing them to say it, that's prior restraint. We review the person and determine that the person at all cannot say anything. That's prior restraint on steroids, is utter violation of the Constitution, but it's not the social media platform in violation. It's the federal government in violation of it. 20 after 7 says everyone's days are numbered. Exactly, man. If you don't stand up, read that poem. I didn't stand up. At first they came for the unjuiced. I said nothing because I was juiced. Actually, it's not even that. I was unjuiced, but first they came for the teachers. I didn't speak up because I wasn't a teacher. I was unjuiced, but then they came for the unjuiced healthcare workers, but I didn't speak up because I wasn't a healthcare worker. Then they came for the unjuiced pilots, and I didn't speak up because I wasn't a pilot. Then, et cetera, et cetera, et cetera. Then out of pressure, I quit my job and started my own business. And every last employee they came for, but I didn't speak out because I wasn't an employee. Then they had the mandate for the self-employed people who couldn't get their tax return unless they got juiced. So I got juiced. And I didn't speak out for the unjuiced because now I was juiced. But then they came, but then I passed by someone. Then they came for the people who had voluntarily put a tattoo on their arm that technologically detects when they've been infected and whether they've been juiced. But I didn't put the tattoo on my arm, so I didn't speak up for them. Then they came for the people who downloaded Apple's geo-tracking system, but I didn't download the geo-tracking system, so I didn't speak up. Then Apple came out with an iOS update that put in the geo-tracking system without my knowledge and didn't tell me about it and waived the consent. And then I walked down the street within 6 feet of someone who had the tracking system and the tattoo, and their tattoo later came positive. And then they came for me. And there was no one left to speak up for me. Everyone's days are numbered if no one speaks up. I mean, pay attention, people. Pay attention. Please. Maybe 20 after 7 says maybe we should call AFOJ. You mean AZ? What's AF? Will says I secretly think this ties to net neutrality. I mean, yeah, all the basis of net neutrality and everything is all precedent to allow, the antitrust and the Section 230, all of whose purpose is not actually to do those things. It's to threaten them so that they can tell big tech who to censor. Ali Reed says NNT number needed to treat, or an NNJ number needed to. Instagram crashed again. So let's see. I'm going to relaunch Instagram. So Ali Reed says NNT or NNJ number needed to juice, NNT's number needed to treat is a better metric to report, not absolute risk reduction or relative risk reduction. I agree that NNV or NNJ or NNT number needed to juice in order to save a death, for example. I agree that is a very good metric to look at, but it is not, it still shouldn't be the default effect because it's still vulnerable to the passage of time. Because if the absolute risk is 5% per year, then the NNT or NNV or NNJ, the number needed to juice in order to save a life, goes down and down and down each year. Whereas with relative risk reduction, yes, that can change. And we know from the fuzzy trial and from the observational data in Israel observing the efficacy of the fuzzy juice, we know that the durability is low and the relative risk does go down. But if you had a stable relative risk, then the relative risk would give you a much better idea intuitively of what's happening over the course of, a 20-year time period because it's staying stable. But I agree with you that that all of these help put the number in context. So relative risk reduction is the default for the effect size that just gives you a very quick intuitive sense of this got rid of half the cases, this got rid of a quarter of the cases, this got rid of 75% of the cases. But then state the absolute risk so you know whether you're protecting against something common or something rare, and state the number needed to juice, so that you have an intuitive sense of like how many people are getting this in order for one person to be saved from something. All of those are valuable, but no one should be pooping on relative risk reduction, in my view. Instagram folks, if you asked a question in the second Instagram that I did not answer, you need to ask it again, because we're now in the third Instagram live after Instagram crashed the second time. So if I didn't answer it before, I didn't see it. John Apagos says, my concern is that our media has no idea what studies actually say, nor does most of the public. How can we convey the meaning with clarity to those who lack the understanding and terminology? Well, I mean, I'm trying to do my best. This was relatively technical because this was, I break something down at a technical level, and then I understand what the proper simplification is, and then I can simplify. So that's what I'm trying to do. Chris Barron says, I like your code. Something like Let's Go Brandon. If you know what Let's Go Brandon is, it's very funny. People were chanting at some race car thing or something like expletive to the president. And the reporter was like, they're all chanting, Let's Go Brandon. And so, people at the last protest on Saturday had giant signs, Let's Go Brandon. I didn't know what it meant, and I asked some people and found out. But then a rapper made a Let's Go Brandon theme song that is utterly hilarious. So go to my Telegram and then look at where I showed a video from Cozy Wellness, Kozi, I think, and click on his profile from the forwarded message in my Telegram, and then scroll up in his profile, and he posted the Let's Go Brandon theme song. If you find it funny, if you find the Let's Go Brandon concept funny, you will be laughing hysterically at the Let's Go Brandon theme song that this rapper made. Anyway. 20 after 7 says, I'm going to donate to your fancy coffee fund, sir. Thank you so much. I absolutely, totally appreciate you. Sherry Thriving says, Dr. Chris, thank you for your time and research and enlightening us. In my opinion, you are a great educator. Thank you so much. I really appreciate that. Sufferus777 says, I have DNA reasons to be anti-juice, like genuine health concerns and survive the virus twice. That just made me think someone should do a cover of Jay-Z's 99 Problems with 99 reasons to get the juice or not get the juice. But anyway, Rebecca Loft says, thank you for your work, time, and effort to share and interpret studies with the public. You're welcome, Rebecca. Thank you for your feedback. Pro-informed consensus, everyone in my immediate family survived with the C word with few symptoms. I have the right to make my own decisions. I have a damn good track record. I've lived 44 years without juices. I support your rights, pro-informed consent, to make informed consent about all your medical decisions. Sapphire777 says, agree, pro-informed, and everyone I know who's had the juice had terrible reactions at first. So what about the long-term effects like farm companies aren't liable for? I can neither confirm nor deny that everyone Sapphire777 knows. I'm not here to make statements about risk right now, but I respect people's ability. I respect people's right to say what they believe, what they perceive the people they know have experienced. Pro-informed consensus, Sapphire777, correct, big pharma is the only people that get immunity from the juice. Duke Man says, Brandon is now no longer the POTUS. It's more like a mascot for the anti-establishments. Well, I once read from Zbigniew Brzezinski that there's always a cycle between strong presidents and weak presidents. And during strong presidents, the president is in power. And during weak presidents, the cabinet is in power. So, that's nothing new. And it makes sense to analyze who's really in power. There's nothing irrational and nothing controversial within political science about the fact that sometimes the president is not the powerful person in the administration. It varies based on the strength of the president. Okay, so I'm going to go to Facebook for live questions now. Oh, I think I've been live streaming on Facebook. My live video ended on Facebook. I didn't know that. Let's see. Can I get the questions and then just let me see if I can pull up the live video from Facebook. If it's posted there, then it might have the questions attached and I can do the questions on Facebook. Uh, I don't see the video on Facebook. Sorry, Facebook, if you asked questions in the live chat. I thought I was live on Facebook, but I guess not. All right, I guess it's time to wrap up. I'll just quickly look at Instagram to see if there's any questions there. All right, looks like we got no new questions. Okay, that's it. All right, thank you guys for. Well, actually, I think I'll just go to last questions. Will says, thanks for taking the time to hear out the audience. You're welcome. Thank you for asking the questions. Quick look at Telegram to see if I did post the link on Telegram and Twitter. So I'll see if there's any comments or questions. No, I think people just came to YouTube from there. And then quick look at Twitter. See if there's any comments or questions. So M Moon says juicy juice. I think people just went to the all right. Oh, so please like, share, subscribe if you found this valuable. And please check out the links for where to follow me on my email list and Telegram in case I go bye-bye. And please, if you got to buy anything that you would have otherwise you go to chrismasterjohnphd.com/support to see my affiliate links. Buy them there, and at no extra cost to you. You're throwing support my way. And then I have information products that are super valuable and underpriced, as low as $2.99. And I have donations for anywhere from 1 penny to $500 billion or whatever you want to give. I don't expect that. I'm just saying that it's whatever you want. So if you could support me in some way, I would deeply appreciate it to keep stuff like this going. I now am going to get off the live camera and I'm going to go work on my juice and transmission article that I'm working with an immunologist on to submit for peer review. And then to simplify down on my blog as a simple message for the masses, and then to make things maximally valuable to legal teams. So I'm doing what I think is most valuable to the world right now during this. So any support you can throw my way while I'm doing that is greatly appreciated. So thank you guys and take care. Thanks for popping in.